This is a production of Cornell University.
So thank you so much for the invitation. This is a great opportunity to grad students to come here and actually talk with professor and grad student and have this like exchange opportunity. And thank you so much, Kenzie, for this opportunity. And thank you so much, Cornell, for bringing me here and talk about a little bit of my master degree or research projects that I'm going to present right now. And welcome everyone. Thank you. So today, my seminars, for today's presentation, my seminar will be covering innovative solutions for soybean production challenges. Today, I'm going to talk about two main projects that I've been working on, which is the first one is evaluating an automated sampling technology for soybean cyst nematode management, and also the efficiency of fungicide Fluopyram against charcoal rot or Macrophomina phaseolina. As a little bit on introduction from the Asian civilization to modern agriculture, soybean has been always used as a nutritional powerhouse and also has been utilized as a catalyst for agricultural farming or more sustainable farming, and originated back in the East Asia like 5,000 years ago until 5,000 years ago until now. It's been also providing feed for human and also promoting agriculture, more sustainable management in general. But today, soybeans are great sources of protein and oil content, so we can use that as a promoting way to have healthy soils, as well as decrease the amount of concentration of gas in our environment by utilizing, for example, a different management as decreasing the amount of tillage right now. So in general, soybeans has been a great impact in our culture on all over the world, and also driving innovation and as I say before, sustainability in general. As a part of my outline, I will cover mainly these 4 topics. The first one is importance of soybean disease management, why we do care about these pathogens and why it's so important to treat them. My second topic will be evaluating automated sampling technology for SCN. This is the first big project I'm gonna be talking about. And the third one is addressing the efficiency of fungicide Fluopyram against isolate of Macrophomina phaseolina, which is charcoal rot, and then as a conclusion, main takeaways. So I ask you, why is so important to address soybean production challenges? What do you think when you heard about soybean production challenges? Why is so important? Any idea?
I like to eat soybeans.
That's right? They're almost everywhere in kind of any product we have right now. Has like some sort of soybean included. That's good. But if you go to more globally, United States as a country occupies in the rank as the second largest producer of soybean. So for the United States economy is actually crucial. As you can see in this graph, I can show you in this graph from this data they have analyzed from 2012 to 2022. United States occupied the second largest producer, as you can see right here. This go and build it up over the years. So just behind Brazil. So it's really important for the community and also to produce profit in general. If you narrow down that with, in my case, in Ohio State occupied the 5th largest producer in the Midwestern area, just following Illinois, Iowa, and Minnesota right here. Ohio itself produced more than 280 million of bushels per acre. That's right here, and it's also really important for the economy as alongside other products or crops. Now we're going to talk about a little bit about SCN and charcoal rot as the top 10 most yield limiting disease. So you can see I took this table from Bradley et al. From Plant Health Progress, and they have been studying all the diseases that affect soybean in general, as well as the production. And they have ranked the soybean cyst nematode as top 1, so you can see right here, in terms of yield limiting disease from 2015 and 2019. And charcoal rot actually occupied the number 9 as the most disease that affect soybean or limited disease. This is basically for North America in general, but more specific as well in the northern states of the United States. If we go back to southern states of the United States, we find also soybean as a top leading yield reduction, and we're gonna find charcoal rot at the 5th one. So these both diseases in general occupy the top 10, being SCN the major one, and charcoal rot has been increasing over the years. I just wanna share this great tool if you're ever interested. This is a crop rotation network disease calculator that you can estimate choosing any disease that affect a soybean, and you can modify them by the years you want to use, and also the disease and the crop, and they give you, or they create this nice table that you might use for your research or your presentation. I just want to share this with you, and you're gonna find the percentage of loss, bushel, and also in terms of economic losses as well. So now we're going to jump off talking about evaluating this automated sampling technology for soybean cyst nematode. A little bit of the soybean cyst nematode cycle. So you can see in this slide, nematodes in general live in the soil. They usually live the surrounding areas of the roots, in this case the soybean roots, and they are usually attracted by exudus released by the plant. And the process of chemiotaxis, they follow the roots and try to colonize or infect the plant. Whenever they arrive to the roots, there's two different key characteristics about differentiating female and female of SCN. The female has usually this lemon shape right here, more surrounded, and the male this like filiform shape. There's a key characteristic of this one. After they start the make process, the female contain all the eggs inside, and the skin of the female it is used as a shell that protects these eggs from the conditions of adverse conditions. One female can have up to 250 on average, so one single female. So that's a lot that you will be encountering in your soil. After that, they kind of follow in the cycle due to factors that are in the environment. They are released, and they will hatch depending on these factors like pH, different temperatures, or there are some stuff that we don't know. For example, genetic like mechanism that we don't know that makes them hatch. So this kind of group these factors. Follow a little bit after they start the first or second juvenile stage. Sometimes they will hatch inside the eggs, and sometimes they will hatch after the eggs. And usually the J2, the juvenile 2 stage, will infect again the plant. And this whole cycle will last around 24 days every single cycle. So how you identify the symptoms in the field? See, this is a great picture that you can find some symptoms of SCN. These chlorotic plants right here, or patches right here, can show you that is like a certain SCN infestation around there. And it's really like visual, but sometimes, or most of the times, an infested field with SCN, we also look like this, like healthy plants. The main reason this occurs is SCN in general, when the population starts to build to above 30%, that's the time that you actually can recognize that in the field. You have less than 30% of infestation, they will remain healthy and above ground, so you are not gonna able to identify that early. So that's why it's so important to do the soil sampling in order to identify these nematodes in your field. So as I say, management of SCN start doing soil sampling, understanding your field population density through soil sampling. And you can start doing by these 3 steps, following these 3 steps. So first, you're gonna collect soil samples, soil cores using a zigzag pattern, trying to cover evenly your field, and then also depending on the size of your field. After that, you're gonna choose from logical areas or management zones. You're gonna take especially care of those like you are using for production of your soybean. So you can see right here. And the last thing to take into account is collect soil from high-risk areas in the field where SCN might be first be discovered, because based on research, SCN or nematodes in general are clustered or a group in a few spots of your field. They're not usually evenly distributed across a field. This is also a really cool figure that why do you need to test your fields regularly? So in this figure, you will find that, let's start with 200x. And we assume that 50% will be 50% will be male and 50% will be males. This female, each one can produce 250x again. So if you multiply that, it will be around 25,000. And if you survive only the 1% of that, and try to build up across every generation, by the end of the 3rd generation, you have around 39,000 eggs in the field. So if you're missing your soil sampling and you're not identifying that early in the beginning, and you might overlook this soil sampling, you are building the population across the generation. And one season can actually have around 2, 3, and to 5 generations and consecutive cycles. So you have seen that soil sampling is really important in order to identify this early in the field. However, this could be really laborious when you have large scale of areas. Also, time consuming. You need a lot of people to do that. And one good thing to mention is that this sampling technology or strategy heavily rely on whole field sampling. They usually do the entire field, and they put all the soil together, and they perform a soil process to just one bag, for example, of an entire field, that 20 acres, for example. So this might be not a good estimation of the population density, in the field. So you might overlook the spatial aggregation as I mentioned in the beginning. So now we're we're trying to use this technology, which is this structure right here. This structure has this specific machine attachment that actually perform the soy sample collection right here. This is new, but it's using right now. It's currently using for do soil sampling, but for edaphic factors in general, like soil texture, like chemical and physical texture in general. So we say, what if we use this to do or perform soil sampling more efficiently and more rapidly? So we're gonna able to identify this spatial aggregation that I mentioned before, doing it more efficiently and also more rapidly. So we base our research question of objectives to first is evaluate the level of agreement between this machine, the automated precision soil sampler, and the manual soil sampling method by determining the number of SCNX per 100 cc of soil. This is a really standard procedure. There is one study before our research project, but they focus on Cyst per 100 cc of soil. What we would see that is we see an issue on that. Sometimes Cysts are not is not the best estimator of your SCN densities in the field, because some Cysts can have full of X of more than 200, and some of them can have 0. So in order to do that, we are trying to repeat this experiment with SCNX. So we make sure we have a more accurate estimation of the field. Once we have that, to prove the efficiency, we're gonna determine the optimal resolution in gridded hectare for soil sampling and detection of SCN population across an infested field. So when we have the efficiency, we wanna say, you need this number of soil sample cores for your entire field to at least are able to visually identify the hotspots in your field. So you can target those specific hotspots with specific management practices. And I want to show you this short video about how this automated machine work. So, take water. So this is one of our soil sampling days. So this is the machine of the implement. It has like a 3-mountain cut behind that actually collects the soil. This is a 10 feet long cut. And there's this mechanism with air pressure that release or detach the soil and insert this in small tubes, cups. And every single cup has their own number and a QR code. So you are gonna able to identify the sample ID and also where they come from. So you're gonna have a specific GPS coordinates for a specific point. So that will help you to, after that, build the maps and so on. So I show you this video. We choose 3 infested fields in Ohio. You can see right here. This was the Western Agricultural Research Station, one of our research stations in Clark County. And the other 2 fields, this small one and large one, was located in Fulton County, more northern Ohio. So this 2 areas has differences in type of soil, as well as climate conditions. For example, Fulton County has more like a sandy soil, so it's and therefore they have higher population SCN population densities. And Ward, for example, have a different and they have a more moderate population density. When we choose our fields, I'm going to explain the methodology we use. So first, we have the soil sampling, which is two. The manual one, we use a probe to collect the soil cores, and the automated machine. Once we have these two methods, we identify the best grid pattern for every single area. One of them being more especially close, and the other ones, because they are large, they're bigger, we tend to use more space grid pattern. So the automated machine makes a cut, as I tell you, 10 feet long, and then on top of that, we're going to zigzag and collect those soil cores. So we are going to able to say that these specific soil sampling came from the same location. So we are able to compare both methods. Once we have this soil sampling, we continue to do the seed extraction. We extract from the soil using several sieves with different sizes. So we are able to collect the seeds. After that, we crush to release the eggs, and using acid fusion as a stain, we use this for, okay, it makes easier to count under the microscope. So we utilize this acid fusion stain that is right here. After that, we proceed to the egg count under the microscope or stereoscope with an X10 focus. And on top of that, after we have our population determination in X per 100 cc of soil, we follow to do the statistical analysis. For this specific project, we use two types. One is least linked concordance correlation coefficient, what is available in one of the packages in our studio. This specific analysis study the degree of agreement between these two methods. So we find this is the best way because actually it gives you the bias, and also it's more accurate, for comparing two methods. And our gold standard is the manual sampling. And after that, we have the inverse distance weighting. We use this special analysis in QRGRS that you input your SCN numbers with the GPS coordinate, and with the inverse distance, it makes preliminary interpolations. So you having your numbers, you will be able to interpolate that and understand the unknown areas of your map. And after that, we do the result interpretation by observing the maps and the CCC value. So this is our 3 locations results in terms of lean CCC. You will find on the y-axis the X obtained by the automated machine, and the x-axis you will find those numbers obtained by the manual machine right here. And if you can see right here, the CCC of the first area is 0.71. So the degree of agreement between these two is 0.71. Being 1, perfect agreement, 0 would be no agreement at all, and negative 1 would be inversement agreement or negative agreement between them. This is considered as a moderate relationship between methods, which is good. But I continue to the second one, we obtain the highest among the 3, which is 0.77. This is the photon 1. And the last one we obtain to photon 2, we obtain the lowest, which was 0.47. But in general, these graphs are telling that these results are really reliable so we can actually perform this automated machine in the future. But at the same time, even though we have moderate CC levels or CC values, after performing the inverse distance weighting with the interpolation analysis, you find with both methods, we are gonna able to identify the hotspots right here, being these red areas right here with the highest number in the population, and this in blue will be the lowest population density. So even though we have these like moderate values, we are able to identify the hotspots in general for both. And something that I want to mention is the how fast this machine can do soil sampling. For example, in this area, we collect 194 sample points, and me and another lab mate, we did this number of soil samples in 6 hours. It took 6 hours. In comparison to the machine, it took like just 2 hours to do the same number of soil sampling. So this is the way we want to treat this to do the soil sampling or collection more efficiently. I want to finish this part. I'm gonna continue with assessing the efficiency of the fungicide Fluopyram against isolates of Macrophomina. A little bit of background of the life cycle. This is a soil-borne pathogen. It's a fungus that usually it's find the first resort of inoculum in the soil. So you can see right here. They usually germinate the microsclerotia under certain temperatures. When they germ, and they used to develop the apressoria and the penetration of the host, in this case, the soybean plant. When they do the invasion, as you can see right here, usually disrupt the water sources, the nutrient transport, so affecting the plant. And the typical symptoms you will find are yellowing, brown leaves, leaves that remain attached to the stems, but this kind of gray appearance. And if you're gonna able to diagnose this fungus makes this really nice and beautiful microsclerotia. If you cut the stem, you will see this microsclerotia forming right here. It's like a small dot right there, and it's easy. And in the field, you will find this more almost daily deadly plant in the field. Kind of the rationale behind this project. This is a screenshot of one of the research I looked for, and it basically gives you the summary of the different management we have right now for Macrophomina. This is from 2021, and we have, even though we have a genetic resistant, I would say genetic resistant growing or developing by companies, we still don't have a resistant variety for this disease. It's just moderate in general. The chemical control, we have fungicide available, but the study that I've read say that sometimes when they perform in the field with fungicides, they're usually inconsistent, or when they are reproduced in the lab is limited, and it's limited with the number of fungicide used. And also we have agronomic practices right here. So as I say, control is limited by the lack of resistant cultivars, and just a few other species are available for crop rotation. Fungicide like the HI, which is usually used for excessively management plant pathogenic fungi, like Fusarium, So we're gonna try to test these fungicides, particularly for Macrophomina phaseolina, because they are not studies related to charcoal rot. So we wanna try to understand that behavior of these different isolates of Macrophomina under the action of this fungicide. And this will allow us to kind of efficiently control charcoal rot in soybean fields, or suggest the concentration we can use in the field. So the objective is assess the fungicide sensitivity for Macrophomina phaseolina isolates on different concentrations. The methodology for this experiment. So we select 10 isolates from Paraguay. We have a large collection from Paraguay and Ohio, and 10 from Paraguay and 14 from Ohio, with the differences. After that, we choose these 8 treatments of the different concentrations, including the untreated one, with 5 replicants of 5 reps. So in total, we're gonna have 40 experimental units, including one by each Petri dish, one Petri dish being a experimental unit. This is where we use PDA as in full strain for the growth of this experiment. Once we inoculate with different isolates, we're gonna start measuring and the fungal growth of this pathogen after 24 hours, 48 hours, and 72, until we have 14 days after application of the isolate. Isolate. We're gonna incubate this in 25 degrees Celsius on a photo period of 12 hours, dark and light, in a regular incubator. After we obtain this diameter measurement, we follow by the statistical analysis, which is estimating the effective control for 50% of the growth, of the inhibition of this fungal growth, with the packages we have right here in our studio. So this is an ongoing project. So I'm still working on that, so I don't have any result to present right now. But I wanna finish with these main takeaways for this entire seminar presentation, which is the comparison analysis show a significant positive relationship for the number of SCNX per 100 cc of soil between the automated sampling and the manual one for these specific 2 locations, Wards and Fulton. The use of this type of automated technology will facilitate the proactive detection of SCN in a field, thereby adding in the strategic development of efficiency management techniques for SCN, and also understanding the behavior of Macrophomina phaseolina under Fluopyram. This fungicide action will enhance our comprehension of the efficacy of this Fluopyram as a seed treatment in controlling charcoal rot of soybean. Is my thank you slide to all members in my lab. This is kind of a picture of my lab in one of the fieldworks. Also, I want to thank my collaborators right here, Integrated Ag team, and all the collaborators. My graduate committee, Dr. Christopher Taylor and Pierce Paul. My sources of funding, like Fulbright, Cal, and also Syngenta, the company. And by that, I'm gonna open the floor to any questions you have or any suggestion or recommendations. Thank you.
I'm curious, you process the soybean samples by hand, is that right? How long does it take to process 100 samples from a field?
100 samples.
I mean, you collected hundreds, right?
Yes. So I would say the first area, we got 194 samples. And it took me, because I did everything by myself, around I would say 1 month and a half. 1 month and a half. Like working like from Monday to Friday, just doing that. Yeah, 1 month, 1 month and a half. Yeah.
How much time of that is counting the eggs?
Just count. Okay. Okay. Right here. So the question was, how long does it take to count the eggs? So we vary depending of your expertise. If you've been doing like so many times, it gets like really fast. You can spend like, I don't know, 2 minutes per sample if you're really fast. And also it depends the number of eggs you're having in your counter dish. Yes.
So how many eggs in one image you count? Like 200 eggs per image or 100 eggs per image?
Okay, so the question was how many eggs they usually count in one sample? We can get from 0, no eggs, until, I don't know, 300 in the Petri.
So you count the 300 eggs to spend 2 minutes for 300 eggs?
Yes, that's in that counter. So we use serial with that, we extrapolate that, multiplying them by 2, because we just count the center of the counter. So we multiply first by 2 to have the entire counter, and then by 20, which is the volume factor we use. So we are gonna able to have the number for 100 cc.
So do you consider about like image processing to like open CV to help you to count the eggs?
Like technology like AI. Yeah. So one of the ideas is to use AI as a way to counter more files. And we have some collaboration with the ag team in Ohio. So it's like ongoing, but we don't have that available yet. Yeah, but that would be a good, yeah. Sure.
Some of your points. So you did like a comparison of the two methods, right? You collect data from 3 fields. 2 of the fields, I mean, the data matched pretty well, right?
Yes.
But the other one, so the data.
The last one, yeah.
Yeah, right. The last one didn't match so well. So what could be the reasons?
So we are assuming that first, that was one of the largest field. So we have like more samples to do. That would be a factor that we have like different variability. And also that area was more towards a pound. There was like a special, like, yes, kind of pound of the farmer. So we are assuming like this might be inducing like different population density. So those are we are assuming that those are the factors that.
So did you notice some like soil type differences? You know, because based on our experience, we noticed, yeah, so your soil types can affect your extraction. So I'm just wondering whether you notice some differences in terms of the soil texture, soil types among those fields.
When we test, we use two soil types, one more sandy soil and the other one, and we got the highest level of agreement in the sandy soil. So we are assuming that even though you have different type of soil, you will get the same value of the same numbers, because we have like 2 examples from different locations and different climate conditions.
I did have a quick question if you have time. Sorry.
Sure. Yes.
Yeah. So what was the rotation history of those fields, and what was your method for ensuring that the cysts you were identifying were in fact soybean cyst nematode and not other species of nematode?
That's a good question. So first, in terms of the crop history, this one of the fields was in research station. They usually have this corn and soybean rotation over the years, as well as the same in Fulton County. They have the same crop history of soybean and corn. And the way we make sure that cysts are cyst nematodes, we use a specific sites of diff, specific site of sieves to extract these cyst nematodes. So we're gonna, yeah, so that's actually the answer.
Great, thank you.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.